Asa Thibodeau, the Jackson Laboratory for Genomic Medicine, CoRE-ATAC, a deep learning model for the classification of regulatory elements from single-cell and bulk ATAC-seq data. Hello everyone, my name is Asa Thibodeau, and first I'd like to thank the organizers for giving me the opportunity to present my research. And so today I'm going to be presenting our recent method, CoRE-ATAC, which is a deep learning model for the classification of regulatory elements from single-cell and bulk ATAC-seq data. To give a little bit of background, single nucleotide polymorphisms exist across the genome and are often difficult to characterize due to being within non-coding regions of the genome. In fact, over 88% of GWAS SNPs are in these non-coding regions, leading to questions as to whether or not these SNPs are dysregulating cis-regulatory elements, whether or not transcription factors are binding to these regions, or what genes these SNPs could be regulating. With the introduction of ATAC-seq, researchers have been able to investigate accessible regions of DNA which could be overlapping these non-coding regions and give evidence that these non-coding regions correspond to specific cis-regulatory elements or potential enhancers. In our previous work, PEAS, we employed a machine learning approach that was based on feature extraction methods in order to predict whether or not an accessible site identified by ATAC-seq corresponded to an enhancer. However, since we use feature extraction methods, we may have missed some features that are hidden within the ATAC-seq data, which could be even more predictive of not only enhancers but other cis-regulatory elements as well. And so our main research question is, can deep learning identify more predictive features from ATAC-seq data? Second, we wanted to understand whether or not these deep learning models can detect cis-regulatory element activity differences due to cell type and/or genotype. And finally, as single nucleus ATAC-seq has become an emerging technology, enabling us to identify rare cell populations via unsupervised clustering, we want to understand whether or not these models can be predictive of cis-regulatory elements within these rare cell populations. ATAC-seq provides us with important pieces of information corresponding to where TN5 transposase binds and cleaves accessible DNA. And these regions can correspond to freely accessible DNA, regions around nucleosomes, or even regions around proteins that are binding to DNA. And so the motivation for applying deep learning to ATAC-seq data is so that these models can learn the difference between cut site and insert distributions, which may reflect the underlying cis-regulatory element function. When training these deep learning models for ATAC-seq data, we focus on three main questions. First, does the model fit the data? Second, is the data diverse enough to capture differences such as read depth and cell specificity? And finally, are the annotations accurate enough to effectively train the model? Here we focus on the first point, on fitting the model to the data. As you can see on the right, we have our deep learning architecture where we combined our previous work PEAS with our novel ATAC-seq data encoders. And the goal of these encoders is to learn new features which we may have missed during feature extraction methods, which should help us better predict the cis-regulatory element function corresponding to these accessible regions identified by ATAC-seq. Each of our ATAC-seq data encoders looks at the center position of regions provided by the user. From this center, we extend 300 base pairs in both the 5' and 3' directions to result in a 600 base pair in total. For the DNA sequence encoder in particular, we are primarily looking at a one-hot encoding, like similar projects in this field. However, we added one slight twist where we look at the ATAC-seq reads so that we can infer the individual's genotype. So for instances where we have enough ATAC-seq reads piling up at a specific position, we then take the frequency of reads corresponding to each of the nucleotides rather than a binary 0 or 1 value for the DNA sequence. Our ATAC-seq signal encoder encodes the 5' and 3' cut sites as well as the insert sites between these cuts in order to get an idea of what the ATAC-seq signal looks like within the 600 base pair window. For instance, we have read pileup information which is analogous to a genome browser profile of this region. We incorporate 5' and 3' cut sites separately, and we also incorporate what the size of this insert is and give a direction by separating out into the 5' and 3' read. Once the ATAC-seq data has been encoded using our sequence and signal encoders, the model then learns different features using 4 blocks of 1D convolutional layers. CoRE-ATAC predicts a total of 4 different classes of cis-regulatory elements, including promoters, which are identified using H3K4 trimethylation, enhancers, which are identified using H3K4 monomethylation, insulators, which are captured by CTCF ChIP-seq, and the other category, which are capturing other histone modification marks, including repressive marks, H3K27 trimethylation, transcribe marks, H3K36 trimethylation, and low signal states where we have an ATAC-seq signal, but are lacking the histone modification mark signal. Going back to our final 2 points for our machine learning focus, we wanted to make sure that the input data for our model training was diverse and that the training labels were accurate so as to not provide the model with misinformation. To do this, we identified over 200,000 genomic regions among 4 different cell types and 6 different samples and/or replicates. For training labels, we used 2 different sets of CoRE-HMM labels, which we corroborated between the two in order to identify a concrete list of CoRE-HMM states that could be used to identify cis-regulatory element annotations. We then took the 200,000 genomic regions and partitioned them into training, validation, and test set regions, where the training set was used for model training, the validation set was used for model tuning, and then the test set of chromosomes 3 and 11 were used for evaluating the model. Using these 2 chromosomes, we were then able to evaluate the model trained on the training chromosomes, and we observed an accuracy of 84.2%. The majority of these mispredictions occurred within the enhancer and other categories. To get a better understanding of the advantages of applying deep learning to ATAC-seq data, and to understand the advantage of having ATAC-seq signal compared to just using DNA sequence, we compared CoRE-ATAC with P's and 2 sequence-based methods, DeepC and LSGKM. Our first key observation is that DNA sequence-based methods perform very similar to one another. So in this comparison, we compared DeepC, LSGKM, and a model trained on just the CoRE-ATAC DNA sequence encoder, and we see that the average precision is very consistent for each of these different methods. We next trained a model using just the ATAC-seq signal encoder, and what we observed with this model is that using just ATAC-seq signal, we get a significant improvement for predicting enhancers just by using the ATAC-seq read pileup information. However, this model trained on only the ATAC-seq signal encoder did not outperform P's, which is likely due to the fact that P's incorporates other information such as transcription factor motifs and conservation scores. Finally, combining both the DNA sequence and the signal encoder with the P's features, we observed that the CoRE-ATAC made an improvement for predicting enhancers even over P's, showing an average precision of 0.92 compared to the P's average precision of 0.9. We next wanted to understand whether or not CoRE-ATAC could predict cis-regulatory function across different cell types that were not used in model training. If you remember, we used a total of 4 different cell types in model training, and in order to test cross-cell type applicability, we tested CoRE-ATAC on 7 different cell types that were not used in model training. Overall, we observed that an average precision of 0.8 for the class average, with promoters being the most highly predicted class, followed by enhancers, then insulators. To understand whether or not CoRE-ATAC is learning a function for predicting cis-regulatory element function, as opposed to memorizing specific DNA sequences, we stratified CoRE-ATAC predictions into regions that were overlapping training regions versus regions that were excluded from model training. And by doing this stratification, we observed that there was no difference between the biased examples versus the non-biased examples. In fact, the only instances where the regions that were biased outperformed the non-biased examples are in the promoter and insulator cases, which makes sense because these specific cis-regulatory element annotations would be more dependent on DNA sequence when you think about the Tata box or CTCF insulator. When we look at enhancers versus others, we see that CoRE-ATAC is indeed learning a function as the excluded regions actually outperform the biased regions. To understand whether or not CoRE-ATAC could predict cell-specific cis-regulatory element function, we stratified the genomic regions of the 40 test samples by their cell type. And the overall observation that we have is that CoRE-ATAC can predict cell type specific enhancers very well, suggesting that cell type specificity is not a limitation of the CoRE-ATAC method. We next looked into individual specificity and how well CoRE-ATAC could identify genetic regions that have variability based on an individual's genotype. To do this, we collaborated with Stitzel and TUI Lab, where they have been very interested in looking at how an individual's genotype can reflect chromatin accessibility. For instance, if an individual has a TT allele, they may have a region that's more accessible than an individual that has a CC allele. In addition to this, they have been conducting massively parallel reporter assays, which enable them to test whether or not that accessible site has an increase or decrease of regulatory activity due to that individual's genotype. Looking through CoRE-ATAC enhancer probabilities for the 19 individual islet samples from our collaborators, we identified a few instances where CoRE-ATAC probabilities reflected the individual's genotype. For instance, for this specific SNP, we identified that individuals with the TT allele had a much higher enhancer probability than those with the heterozygous allele, which had enhancer probabilities closer to 50%, and individuals with the GG allele, which had a much lower enhancer probability overall. We then separated all instances where CoRE-ATAC probabilities reflected predicted loss or predicted gain based on an individual's genotype. And then we compared those instances with gains and losses from MPRA log fold change values. And this showed that the MPRA log fold change values were concordant with the predicted loss and predicted gain values from CoRE-ATAC, suggesting that CoRE-ATAC has some evidence of being able to predict an individual's specific cis-regulatory element activity. Moving on to our final part of our talk involving single nucleus ATAC-seq data. So in the advent of new technologies such as 10x Genomics Chromium platform and Cell Ranger ATAC-seq software, researchers have been able to perform ATAC-seq in single cells. And the key advantages of single nucleus ATAC-seq are, one, it enables the integration of cis-regulatory elements at the single cell level, and two, we can now profile the accessibility of rare cell populations that we otherwise couldn't be due to lack of cell surface markers via unsupervised clustering. With CoRE-ATAC, we focus on the second point, where we want to investigate whether or not CoRE-ATAC can predict cis-regulatory element function in these rare cell populations that we can identify using unsupervised clustering of single nucleus ATAC-seq. So to test this, we obtained public PBMC single nucleus ATAC-seq data and performed unsupervised clustering to identify a diverse set of immune cells. Using these immune cells, we then obtain chrome-HMM state profiles in order to provide us a list of annotations associated with these different accessible regions corresponding to these different immune cells. And what we observed using CoRE-ATAC is that we still maintained a high average precision for predicting cis-regulatory element function, despite there being differences between bulk and aggregate single nucleus ATAC-seq background profiles. So this suggests that we can use CoRE-ATAC in order to interrogate these rare cell populations and to further study these different cell types that we otherwise couldn't have before. Finally, we investigated CoRE-ATAC's ability to identify important enhancers from both single cell and bulk ATAC-seq data. And we observed that the majority of super enhancers within these different cell types were being recapitulated by CoRE-ATAC enhancer calls. Furthermore, when we investigated the GWAS SNPs at these enhancers corresponded to relevant diseases within the cognate cell types, suggesting that we can further prioritize enhancers that are relevant for disease. In summary, we trained a deep learning model that effectively learned differences in ATAC-seq signal and DNA sequence to predict the functional annotation of cis-regulatory elements. CoRE-ATAC can be used across different cell types, is genotype aware, and is a valuable resource for detecting cell or individual specific cis-regulatory element activity. CoRE-ATAC can be broadly applied and extends to aggregate single nucleus ATAC-seq data, enabling the interrogation of cis-regulatory element function in rare cell populations. If you want to find out more, you can visit our bioRxiv paper, or if you're feeling a little bit more ambitious, you can try out our code by visiting our GitHub page. I would like to thank the Uchar, Stitzel, TUI, Lee, and Beck Labs for all their help and feedback throughout this project, as well as the Farm Foundation and NIH and IGMS for funding this project. Thank you.